So it gives me great pleasure to introduce Dr. Michael Cameron. He's a professor of molecular therapeutics and the director of the drug metabolism and pharmacokinetic branch within the Translational Research Institute of Scripps, Florida. He received his PhD in biochemistry from the University of Utah. He has worked in the overlapping fields of drug metabolism, molecular toxicology, and molecular optimization, driving research studies to better understand the influence of metabolism on toxicology and the physiological protective mechanisms that mitigate that toxicity. He has designed and conducted studies for both large and small pharmaceutical companies, evaluating potential drug interactions and identifying reactive metabolites. They may lead to toxicity in over 100 compounds in clinical trials designed to support investigational new drug and new drug applications to the U.S. Food and Drug Administration. One of the drugs he has investigated in the past currently ranks in the top 10 of the prescribed drugs in the United States. Dr. Cameron's World Trade Center Health Program research is designed to lay the foundations to optimize the clinical use of a non-electrophilic Nrf2 activator in slowing or preventing the progression of pulmonary fibrosis, thereby potentially extending lifespan in individuals with a disease that has no current cure. It gives me great pleasure to welcome Dr. Cameron. The floor is all yours.
All right, so let me share my screen. So as was introduced, this was a start on a drug discovery project. So we're looking at one of the protective mechanisms that is innate within humans and seeing if we can turn that on and if that can then have a benefit. So when we just talk about just in our regular lives that there's a constant, you expect to do well, and you hear about such and such is doing bad, this is going bad. And we kind of come across almost thinking that we're maybe a little frail, that we're not really that well designed. And there's a ton of different ways that things can go wrong. And that's not actually very close to the truth. We're actually quite well designed. We can have rather amazing sort of insults and we can come out on top from that. That this cute little baby will probably become this happy gentleman just given time. And he's going to come across thousands of chemicals and thousands of bacteria and different viral challenges. And that's probably just what happened yesterday. That's not even talking about what's going to happen in their lifespan. And that this is something that the existing mechanisms that we all have, that they need to work. And he needs to essentially win the fight against this bacteria or against this virus again and again and again. And there may be a few times where you have to have some antibiotics or you have to have some treatment, but by and large, we actually do win that fight again and again and again. And even if you were to go back, what's really sort of amazing on this is this little baby probably isn't that different genetically from a little caveman baby. But there's entire new environmental chemicals. We've had all sorts of things like COVID came up and we see all these different mutants. These weren't even around when caveman baby would have been around, but you have to have mechanisms that can address all of these, that can manage all of these sort of problems and can respond to them and keep this young kid safe so that he grows healthy and happy. And I know we probably all remember the whole thing about COVID and we can just maybe give a bleach or strong light or something. And that's a terrible idea. That if we tried to give something like that, frankly, we're just not very good at it. That we would bathe not just the cells that are having a problem and the cells that are infected, but we're going to bathe all of this healthy tissue with an incredibly strong insult. And we're going to cause far more harm than we were going to have good. From you being the one that is administering this, this is a terrible idea. But your body does not think it's a terrible idea. Your body thinks it's a pretty fabulous idea. Your immune system, one of the cells that's involved in infections is called a neutrophil. And a neutrophil, in here you see these little tiny round blobs. They refer to these as granules, and the granules are filled with stuff that should be kept away from the rest of the cell. They're kind of like a little Tide Pod where you've got something that's contained inside, and then when it's needed, it goes to the surface, opens up, and spills out into the extracellular environment. And this neutrophil moves directly adjacent to the bacteria or directly adjacent to the infected cell. And one of the things that it secretes out is hydrogen peroxide. And this is the exact same hydrogen peroxide that your mom probably used on cuts and kept for different bacteria to keep you safe. Well, your body uses the same thing. It also secretes an enzyme called myeloperoxidase. And myeloperoxidase takes hydrogen peroxide and chloride. This is just table salt, and it catalyzes it and turns it into bleach. So you providing bleach to the system is a really bad idea, but your body thinks that bleach is a really good oxidant and that it's very non-specific, and all it really has to do is be able to identify what's foreign, what's diseased, what does it need to remove, and then it can use this very non-specific warhead and send bleach right at the surface of exactly where it needs to be, instead of spilling it over all these healthy tissue. That there's a difference between we treat the entire organism versus we treat sort of where it's needed. And this goes on with say, cancer, that your body does not just say, well, came down with cancer and just move on from there, that it actually fights this, and it's fought this again and again. And chances are for most people, it's won that fight multiple times, and that you've had multiple cells that have actually become cancerous and that they were cleared out by your own immune system. This has brought on the whole field of immunotherapy, which traditionally we treated cancer by just pumping somebody full of poisons. And you basically went on the assumption that if you beat up the whole body, that perhaps those healthy cells will be able to withstand this insult, and the diseased cell, it'll push it over the edge and you'll get this cell to die. But in here, there's a science article talking about cancer immunotherapy, and you can read this whole thing, but I've summarized it down into one little blip at the bottom, which essentially says that your body can customize a defense, that you can come up with very specific antibodies that recognize very specific antigens that are either at the surface of the cell or the surface of, it doesn't have to be cancer, it could be a different infection or whatever your incidence, that you can design a response to it. And in the Nature article, this is really an exciting area in cancer. It's the immune checkpoint. And what they're looking at is, can you boost the defenses that your body is already deploying? And this is very much what we're looking to do in the project that I'll be discussing in a little bit. And within these, your body will try to fight the cancer. And the healthy cells with immune cells, sometimes they need to just dampen them down a little bit. That you don't want inflammation, just chronic inflammation. So there are specific ways that you can sort of silence an immune cell. And a cancer cell sometimes can recognize how it can do that, and it can silence the immune cell to have it where it's not functioning the way it should. So in this is just one example of a checkpoint inhibitor. It's programmed cell death protein 1 and ligand 1. And you have your little T cell that recognizes the cancer cell, but the cancer cell can turn this off and then it can grow. Where if you provide the appropriate checkpoint block, a lot of times these are specific antibodies or small molecules that turn this on or change the way they're presented. And then the tumor cell is not able to turn off the T cell, and the T cell can kill the cancer cell. And you can attack this from both ways. You can protect the T cell, or you can take away the tool that the cancer cell is actually working. And this has opened up entire fields that we didn't have just a few years ago. So what's this got to do with anything that we're talking about? So World Trade Center has associated pulmonary pathologies, and many of those pathologies have an oxidative stress or an oxidative stress injury component to those. And the hypothesis was that we can activate immune protection, innate protection mechanisms that were designed to protect us, and that we can utilize those instead of sort of starting from scratch. We'll use what's already there and build on what's there, turn it on, help augment the response that we've already have. So the system that we chose to work with was called Nrf2. So Nrf2 is called nuclear factor erythroid 2 related factor 2, which is really big and it kind of messes with the flow of the talk. So we'll just call it Nrf2. But it's the master regulator of oxidative stress. With response to oxidative stress injuries, you usually upregulate Nrf2, and it causes a lot of cytoprotective genes to suddenly be expressed. That it's a transcription factor. We'll go into what that means, but it has a couple of areas. It sequesters iron. Iron spins off a lot of free radicals. It increases the reducing potential within the cells. These multi-drug resistance associated proteins, these are efflux transporters. They essentially kick things out of your cell. And this is one of the very large ones, the glutathione metabolism. Glutathione is one of your body's main antioxidants. And upon activation of Nrf2, you get a significant increase in the cellular concentration of glutathione because it upregulates the different genes that are responsible for making glutathione. So within this project, we're looking for a non-electrophile activator. What's this mean? Why would we care? So an electrophile, electro is for electron and phile is a Greek root for loving. So we've got exactly that, it's electron loving. So electrophiles are electron deficient molecules, and those electron deficient molecules can go and can pull those electrons from existing biological proteins, biological small molecules, but you can then have covalent bonds formed between your electrophile and these biomolecules. And we've probably, many of us, seen at some point the different amino acids. And you see all these different amino acids within proteins, and they tend to classify them as these ones are acidic, and these ones are hydrophilic, and these ones are polar. And they throw on a couple other different classifications, which ones are essential versus non-essential, essentially which ones can you make versus which one do you need to pull in from your diet. One thing that we don't really talk about is which ones are very susceptible to electrophilic attack. So cysteine and lysine, this nitrogen at the end of the lysine and the sulfidyl group at the end of the cysteine are very electron-rich parts of the molecule, and they're very easy to oxidize. And when they oxidize, you actually will end up with a modified protein that no longer has just the free cysteine or just the free lysine. You can have the lysine and whatever oxidized it. So you're fundamentally changing the protein. And that's what we didn't want to do. We didn't want to say that we're going to go in and we're going to look at something that's an oxidative stress against the oxidative stress system, and we're going to do that by damaging the system. That we're going to cause oxidative stress and hope that the response to that is sufficient that we end up with a net positive. So, sorry, this project was pretty heavily interrupted by COVID, and I tested positive yesterday. So, but I'm feeling okay. Under good conditions, we stay in this green area. That this area that we see here is a ubiquitin lipase system, and Nrf2 is the protein that we're targeting, and it will go to the nucleus and cause a lot of the expression of a large number of genes, or transcription of a large number of genes. But under most cases, you end up with ubiquitin added to the Nrf2. It ends up with multiple ones called polyubiquinated, and then this goes to the proteolytic degradation and it chews it all up and you start over again. But if you end up with oxidative stress conditions, you end up with these little cysteines that are sitting along the ligase, and they get modified. And when they're modified, they don't pass on the ubiquitin to Nrf anymore. Nrf levels rise up, they go to the nucleus, and then they start upregulating a whole bunch of different genes. So the issue with this is that there's nothing specific about this cysteine versus the cysteines in many, many of your other proteins. So there is absolutely no reason to believe that if you were to give an electrophile as your way of activating Nrf2, that it's truly just activating it here, and it's not messing with protein A and protein B and protein C, that under those sort of conditions, you're interacting with a whole lot of proteins, not just the ones that you maybe wanted to interact with. So we started this off with a high throughput screen. It had a luciferase reporting gene. There were a very large number, over 400,000 compounds. Of those, there were 1,200 hits, 800 of those confirmed, and a secondary assay using a different readout had about 380. And this isn't actually that less than half confirmed. Some of this was that in the secondary assay, there was a full dose response. And in some of these, we just had so little compound remaining that we couldn't actually test them in the secondary assay. But we had 388 active compounds from the secondary assay. Of those 388 compounds, 28 looked like they were non-electrophiles and that they worked by some mechanism other than covalently binding the protein. Because these compounds had been screened in so many assays, we could look at these 28 and say how many things had they been hits against before. We could also look at how potent they were. There were a couple of them that had some levels of cytotoxicity. There's a couple of them that looked like they were really, really difficult to synthesize. And it kind of left us with two that we were most interested in. So those two compounds were what we moved forward into this exploration. And what we wanted to answer were two key questions. Will these compounds activate Nrf2 in a relevant cellular model or in vivo model, not in a cell-based reporter assay? And how do they work? So we started off by verifying that they weren't nucleophiles. We didn't think they were based on their structure, but we ran a quick little assay and verified that they weren't a nucleophile or an electrophile. I don't know why I wrote nucleophile, that's wrong. And to verify that they were not metabolized to an electrophile. And it passed both of those. We also checked the solubility. These numbers aren't particularly good. It's pretty insoluble, but it wasn't tragic. What we found out later was that this is a little misleading, that we didn't have compound that went from soluble to not soluble. We ended up having a lot of aggregation. So you would get one of the molecule that would come together and you'd have 2 and 3 and 4, and they would sort of stick to each other like little glue. But they didn't stick with so many of them that they actually fell out of solution, but they started behaving a little different. And this turned out to, for some of the things, it was a problem. We were able to work around it in some cases, but the solubility ended up being a major problem for these. We also looked at their metabolic stability, and these numbers for human are okay, but for mouse, this says that this compound is going to be degraded pretty fast. But we weren't as worried about this because of the nature of what these are. So we initially just dosed it and did a PK study and said, we're going to dose these two compounds and we're going to look at what the plasma concentrations are over time. And we're going to dose it at 3 different levels, see the plasma concentrations over time, and you can see by 6 hours that it's really dropping down. By 8 hours, there probably wouldn't be anything detectable anymore. If you remember the basic central dogma of genetics, you go DNA to RNA to protein. And that's sort of what we were hoping was going to help us, that we're getting these molecules that are going to direct something to the DNA, and that's going to make RNA. And the RNA has its very own half-life. And a lot of times those are in the 8 to 24 hours. And then those make proteins, and those proteins have their own half-lives, which can vary massively from minutes to days. So what we were hoping was that even though these compounds don't actually stick around that long, was that the RNA and the proteins that they code for will stick around longer, and that we can get a durable response, even though the compound's not actually there that long. So this isn't actually a transcription factor, this is a nuclear hormone receptor, but it gets the same point across, that a small molecule comes in, interacts, that from that point, you get a shuttling to the nucleus. From the nucleus, you get RNA synthesized that gets pushed out and makes protein. And that we were sort of banking on this RNA portion and this protein portion sticking around long enough that this is actually something that we can use for a therapeutic. And then from there, essentially what I've been saying, is it possible to have benefits of Nrf activation after the compound may be gone? So we looked at target genes. So this NQO1 is a gene that Nrf2 causes to be synthesized. And we looked at 6 hours after, what are the levels of, so this is actually from the study right up here. So at 6 hours, these mice were euthanized, and we used their lungs to look at these protein levels. So we were able to say, what is the level of these mRNAs, and show that it had actually increased it in a pretty dose responsive way, that the higher doses gave higher amplification, the lower doses were lower. And we went back through later and said, how long does this stick around? So let's look at it at 8 hours, 16 hours, 24 hours. And it certainly is higher at 8, but we are seeing the mRNA itself that's sticking up even at the later time points. We also wanted to verify that this wasn't just, these were done in male mice. We wanted to make sure that there wasn't a difference with male and female. So we looked at the amplification as far as the upregulation of the mRNA in male and female mice, and they were the same. So that was nice. So then we were getting ready to do a whole bunch of in vivo studies, and then everything got closed down. So we were just completely stopped for a couple of months. And then even once we were back up, it was a situation that you could work in the lab, but you had to keep social distancing. So how do you do these certain studies? And there are some studies that work better for social distancing than others. So that sort of pushed us a little bit as to what we chose to move forward. So mouse efficacy studies, which is what we really wanted to do, essentially got put on hold. And we refocused to in vitro experiments to demonstrate the activity in human cell lines and to examine the binding site. So we started off by getting some human bronchial epithelial cells, and from these bronchial epithelial cells, treating them with the Nrf2 activators to see if we could get an increase in the mRNA in an actual human cell line. Where we had been doing this earlier stuff in mouse, obviously we're not looking to make veterinary treatments. So it's important that the same thing works in human as well. And we were very happy that we do see upregulation of 2 different target genes for Nrf2 when it's pretty dose responsive in a human epithelial cell. So we went from this, which is looking at mRNA, to the next one, which is looking at actually a protein level. So we were looking at NQO1 is an enzyme. So we were looking at the enzyme activity within these cells and tested two different compounds. One is a GSK control that I'll talk about in a little bit, and then our compound. And you can see that we start to see an upregulation in here at much lower concentrations for the compound that we were looking at over the GSK control, but perhaps that they get a little higher upregulation at the end, but this concentration is, they can't actually achieve in vivo. So take it for what it's worth. We also wanted to go one step further and say, now we've done this with mRNA, we've got this with protein levels, and what is it with a small molecule? So from there, we were looking at actual glutathione levels, and you can see within the cells that as you treat with higher and higher concentrations of the test drug, that you can see an increase in the baseline glutathione levels within the cell. And these ones were already starting off fairly high. This micromolar is actually when we lyse the cells, so the concentration within cells is much higher for glutathione than 5 micromolar. But we can see about a 40% increase. Under diseased cells, you start off with much lower levels. So in a cell that's experiencing a significant oxidative stress, this level would be dropped about 50%. And being able to induce it back to its normal levels has a huge impact on cell survival. So we also started looking at, can we see where this goes? So this was a strategy, and we wanted to say, here's the hit compound, and we wanted to add on a little bit to it that had a photoactivated probe. So this is the photoactivated probe. In response to blue light, you get a rearrangement of this, and this gives you something that's very reactive and will covalently bind to the protein. And then this alkyne here acts as a little fishing pole. There's a technique called click chemistry where you can click that onto biotin, and you can pull the proteins out that have this with it. So we were looking at saying, can we get these probes and figure out where this is actually binding to the protein? And what we found was that we started off with our initial probe, and the probe wouldn't upregulate Nrf2. So then we made a little longer probe, a longer linker here, and that wouldn't upregulate. So then we went to a couple other different sites on the surface, and those wouldn't upregulate. So in the end, we weren't able to get a compound that actually bound to the protein in a way that was active. And we didn't want to do a huge project that ended up saying, well, we found these spots that it binds, but we don't actually know if that's the way it really binds in vivo because it doesn't work. So we decided to go for a different mechanism. So this was our main strategy. We ended up going with a backup strategy. So Nrf2 binds to this protein called KEAP1, and it binds right at the surface. And there's a known interaction. There's a specific part of Nrf that binds with this KEAP. The top end of the KEAP1, they call it a beta propeller. It's a whole bunch of beta sheets that come together in this little bundle, and they make like a little core down in the middle. And if you overlay the actual peptides that are within Nrf2, there's two ways that it sort of fits in the active site. And the base idea is that if you take a molecule that binds in that active site, that you can physically take up the space, and then this can't bind in there, and that you can cause a dissociation of these two proteins. And when you have them dissociated, it's not going to get degraded. And because it's not getting degraded, the concentration goes up, it moves to the nucleus, and you end up with higher levels of the target genes made. So we decided to test this with a fluorescence polarization assay. So fluorescence polarization has to do with the time frame that fluorescence takes place and the time frame that molecules rotate. And essentially, you start off with light, and light comes, it's a wave. And when you just shine a white light, that it's very disordered, and you have that wave going in all these different directions. You can pass it through a filter, and you can end up with polarized light that's all going in one direction. And if you've got, in this case, what we had was the peptide for Nrf2, and we put a fluorophore on the end of that, that if it's on something large, this moves very slowly. So it absorbs the light that's got this very specific polarization, and then it fluoresces with the exact same polarization. And you can detect it by saying, between this and my detector, I'm going to put another filter, and it's either going to be in the same direction as the way that I shine the light, or it's going to be exactly opposite. And if this is bound, it's moving slowly, and you end up with almost all of it in the same direction and very little going the other way. But if it's not bound, this little tiny peptide, it's not very big anymore, and it rotates a lot faster. So your compound comes in, binds, kicks this off. When this polarized light comes in, it is coming out as fluorescent, but it's rotating faster than the fluorescence is coming off. And because of that, it jumbles everything up. And when it jumbles it all up, you see light, you detect it not just in the direction that you had initially done the polarization, but you detect it in the other direction as well. So this was a strategy that we took. So we started off by developing our assay and saying that we were going to take this amount of fluorescence polarization, and we were going to titrate in more protein. And it worked. As we put in more and more protein, we get more and more fluorescence polarization, because more and more of the peptide is actually bound to this big protein here, instead of the peptide floating around by itself. And then we wanted to see, can we inhibit this by then saying, let's just put in more of the peptide, but we're going to use the peptide that doesn't have a fluorophore. So it's the exact same peptide that's going to come down, and it's just going to take up some of those binding sites. So we're going to have a higher and higher percentage of the actual labeled peptide is going to be free, because non-labeled stuff is taking the place. And we see a nice decrease in the fluorescence polarization, and this is exactly what we would think. So then we looked at two compounds. One was a compound from GSK, and I'm going to swing back up to this one, because this is kind of an important part, that when you look at this edge in here, that it's kind of hard to see, but when this labels the actual amino acids, and it's N382, Y334, S and Y, and you get all these different things, and it's mostly YSRNDE, which are all polar amino acids. All of these have an oxygen or a nitrogen right at the surface on their side chain, and are involved in hydrogen bonding. So a molecule that's going to come in and going to bind in this tends to have a lot of polarity to it. And in almost all the cases that they've had molecules for this, most have had a couple of carboxylic acids, and those don't make for the most permeable of molecules. So we were looking at our molecule and thinking it looks nothing like those. So maybe it binds in a different way. What we thought was that maybe it would bind in between some of these beta propellers and sort of wedge it out a little bit. But what we ended up seeing was that the compound that we knew bound right at that interface did exactly what it was supposed to. And the fluorescence polarization decreases as we titrate in the compound. And the compound that we were looking at doesn't show any interaction with it at all. So it looks like at this interface here that We still don't know what the mechanism for this is, but it's not that it binds at this interface and prevents these two from binding to each other. That's not horribly surprising, considering that in this case, you're binding the native case, you bind an electrophile to these sulfhydryls, and none of these are actually close to this binding interface. They're much closer to this area in here, which may be involved in the overall orientation of this, because the distance between the molecule up here and this ubiquitin is quite important for it passing on the ubiquitin. And it's possible that it binds down in some other spot down in here and interrupts the this passage of the ubiquitin. But in the end, we weren't able to find that out. So as we ended up with COVID restrictions lifted, we went to one of the major pulmonary fibrosis models. This is a model that's used in virtually every drug discovery project for pulmonary fibrosis, because it's very reproducibly causes pulmonary fibrosis in a short period of time. So in this case, we're looking at bleomycin. Bleomycin is an anti-tumor, it listed as an anti-tumor diabetic. It's actually isolated from a soil bacteria, a streptomyces, and it's used for treatment of multiple forms of cancer. And it has two mechanisms that it works. So one, it just binds to DNA. When it binds to DNA, you have a hard time of making a copy of that DNA and having the cell divide to where you end up with one cell becoming two cells. So that's one of the important things for cancer cells, that they like to grow. And if you can interrupt this growth, that can be a benefit. The other thing is that it binds iron in a portion of the molecule and spins off oxygen-free radicals directly adjacent to the DNA. And this causes DNA strand break. And they think that this high oxygen exposure in the lung is the reason why it tends to be more toxic. It's toxic out to all the cells, but it's particularly toxic to the lung. And they think it's because of the high oxygen concentration within the lung that you get way more of these free radicals, and that it's causing way more DNA damage. There's a lifetime maximum of how much you can have of bleomycin, where they just start to say that the risk of pulmonary fibrosis is so high once you get above this level that we just can't give you any more of it. So we were using this drug to induce bleomycin, pulmonary fibrosis in mice. So this is after we give the compound by oropharyngeal dosing. So that is essentially where you put the compound right in the back of their throat and you close their nose for just a second, and they pull it in, and it pulls the drug into their lungs. And we found that this actually dispersed the compound within the lungs much better than if we used like a gavage needle and pushed it in ourselves. But you can see within a healthy lung that it tends to be fairly uniform in color, that it's pretty smooth, and the edges have really defined edges. There's nice corners on these. And in the bleomycin treatment, we lose those edges. They round out. We're getting these banding where it's almost contracting upon itself. It almost looks a little like a brain now instead of like a lung. And you're starting to see areas that are a little whiter in with the pink. And it definitely, you're seeing a response from fibrotic response after dosing the bleomycin. When we took this and looked at the histology on a nice healthy lung, you can see this like snowflake-like lattice, that it's got big areas in it that allow for oxygen exchange. You breathe in, the oxygen comes into these areas, and it exchanges within with the capillary beds. And when we looked at this after the bleomycin treatment, we see an entirely different look. The same tissue is much denser, and the stain, the connective tissue, is in blue. We start to see a large increase in the amount of fibrotic tissue, and which probably isn't surprising given the banding that we can see a physical difference in the lungs. But you also start to see that there's not these little holes, these open spaces. And that's because we've busted down the barrier integrity of these open spaces, and they've filled up with different immune cells. They've filled up with just red blood cells that have spilled into the space, that you're now filling up the lung essentially with blood. So we looked at this a couple different ways. This is vehicle versus bleomycin. This is for collagen 1A1 and collagen 3A1. So these are actually the protein that makes collagen, that makes for pulmonary fibrosis, you have fibrinogen. And this is the collagen that's making the matrix that's causing your scar tissue within the lung. IL-6 is a pro-inflammatory cytokine. In this case, we saw a large increase in the pro-inflammatory in the first 7 days, which largely seemed to resolve itself by 28 days. Here, TGF-beta, it's another inflammatory cytokine. These two are total protein and LDH. So total protein, this is from lavage fluid. So took the mice, pushed a small amount of saline into the lung, pulled that back up, and did that a couple of times, and then looked to see how much total protein do you have in the treated group versus the vehicle group versus the bleomycin group. And this is largely a view of barrier integrity. So you shouldn't have a whole lot of protein in there. And what we're seeing is a breakdown of the barrier, and that's causing the difference in total protein. And LDH is an enzyme that's in a lot of places, but you can also see it in the plasma. So it shouldn't be at high levels in the bowel fluid. We also looked at 4-hydroxyproline. 4-hydroxyproline is a modified amino acid that's part of collagen. So here we don't see a huge increase on day 7, largely I think because it's just starting to lay these down on day 7, that the mRNA is up, but we haven't put down a huge amount of collagen yet. But then that's continuing over these next 3 weeks. And over the next 3 weeks, we start to see an increase in the total amount of collagen laid down within the lungs, which you can really see in here with these nice blue staining within the lung tissue that wasn't within the original healthy treated lungs. So bleomycin injury, dosing bleomycin in mice, it was giving us very reproducibly pulmonary injury. It was giving us fibrosis. And we wanted to know, would a non-electrophile Nrf2 activator protect against bleomycin-induced pulmonary injury? So we were really excited, and we started the study, and all the mice died. The problem was that the mice didn't just die in the compounds that had the Nrf activator. The mice died in the vehicle group too, and they died at the same rate. So we played around to see initially a response A, can we change the formulation? And we tried a bunch of different vehicles. Initially, we'd started off with something that had a fair amount of DMSO and cremophore and saline, and this is because the compound's not very soluble. And it took a fairly aggressive formulation to get it in solution. We tried multiple different vehicles, didn't find a whole lot that was more gentle that would get it into solution. We did try to dose a couple of the suspensions, and they didn't work. So we ended up with very low plasma levels. And because of that, we wouldn't expect to have high levels of increase of the Nrf2 target genes. We tried to dose orally, and it was minimally orally bioavailable. And our next response, this was a 1-year project, and we were really running short on time. We had already asked for an extension because of the COVID pushback. And we decided, let's change the model. So we had been doing this in Bleomycin. Bleomycin may be something that's used in the pharmaceutical industry, but it doesn't actually fit with World Trade Center. Nobody in the World Trade Center incident actually came across Bleomycin. So we wanted to test this. What if we switched to an asbestos model? So asbestos fireproofing was used in World Trade Center, and it was a component within the dust cloud and debris. It's little fibers off of rocks. So I've seen numbers that vary drastically as far as how much asbestos was in the different dust. And they're probably all right. It's that this is small and it moves around and it can move in the air, but it also settles a little different than different other particulates. And sampling and getting a representative sample is probably very, very difficult. And you're going to end up with a lot of variability based off of that. But these individual types of asbestos, we use the acrositile, and it's sprayed onto the supports. I read someplace how many floors up they spread it to, because there were changes in the rules, and above a certain floor that they switched to a different system rather than asbestos, but I don't remember what that level is. But this was a study that we did. So we used C57 black 6 female mice, 12 weeks of age, and dosed with either vehicle or asbestos at 250 micrograms by oropharyngeal dosing. And we dosed on day 0. On day 7, we did mouse ox, which doesn't probably mean anything. It's a trade name, but we had done a previous pilot study where this is something that measures the oxygen saturation, and it measures their breath rate, and it measures their heart rate. And we had seen that when we did this in the mice, when we did a quick evaluation of the asbestos, that we were starting to see functional deficits, that the mice actually weren't breathing as well. They were having lower oxygen saturation, and they were compensating for that by having a higher breath rate. So they were breathing faster so that they could keep their oxygen levels up. The problem was when we did this study and we brought it to a bigger, and we tried to use an intervention, that didn't replicate. And I don't know if it's just a matter of variability, or if maybe it was a little bit that the asbestos size or slight differences in how we did the oropharyngeal, that they sucked it in a little better on the one time. But we looked at the cytokine array and then the mRNA analysis and histology at day 35. But the one thing that we didn't see is that we didn't see a functional deficit by day 35. The mice still had normal oxygen levels. They were breathing at a normal rate. When we looked at the histology, the lungs looked pretty similar. So I think if we were going to do it again, we would either give multiple doses of the asbestos, because people that were coming across asbestos in the regular environment is usually an occupational thing, and you don't just come across it once. So we would probably do repeat dosing a few times, and I think we need to run it out longer than 35 days. But we did see something that was kind of interesting, and this was that the Nrf2 genes, asbestos upregulates them too. So asbestos is an insult, and the mouse is utilizing existing protective mechanisms within itself to deal with that insult. So we didn't have any problem with the vehicle, but we see upregulation of two different, and we have some others too. But these different Nrf2 target genes, the asbestos treated animals were all higher. In all cases, when we treated with not just asbestos, but with the Nrf activator, we did see an increase to that. And we weren't sure if that was going to be enough to actually cause a functional benefit or not, but we were interested in seeing that. So, as I said earlier, the histology and pulmonary evaluation didn't show impairment at the end of the study. But we did see mRNA levels of collagen proteins were increased in the lung after asbestos treatment. So we are seeing this COL1A1 and 3A1, and then also this ACTA-A2 mRNA. And this is a mRNA that's associated with myofibroblast association, which is essentially one of the cells that lays down scar tissue. So we're starting to see these upregulated within the asbestos treated group, but it probably didn't run out long enough to actually have a functional deficit. But we are seeing that in response to the asbestos, that there is a cellular difference for in those treatment groups. And the treatment with the Nrf2 activator, in all cases, did bring it back towards the mean. So it doesn't look like we brought it all the way back for any of the groups, but it is lower for these different proteins that lay down collagen or fibroblast-associated protein. And I think if we were to run this out a little longer, we might actually see sort of a true evaluation of how well does this work. So I think this is very encouraging. We were just right up against the clock and running out of time on this. So future studies, I think that we need to extend the window and maybe do multiple exposures to see, can we make a functional deficit? So can we actually get damage to the lungs, and then can we protect against that damage by the Nrf activator? So conclusions, the non-electrophiles can be used to activate Nrf2 in both human and mouse cellular models. The activation is observed at the mRNA, the protein, and the small molecule levels. That the half-life of the mRNA and the protein are longer, and this has helped the compound in the functional assays, even though the compound doesn't have the best PK. Really, we would like to increase the solubility and decrease the aggregation properties of the compound. So we would like to play around with a few new analogs to see if we can come up with maybe a little better compound. That this was limiting. I think the Bleomycin model probably would have worked if we didn't have to bash it with such an aggressive vehicle. That activation uses some mechanism other than blocking the KEAP1 Nrf2 interface. So pharmaceutical companies are coming at it from this angle. They're trying to block this interface. And we haven't figured out what the mechanism is, but it's unique and not going through interfering with this interface. And we think that because this molecule here is considerably more hydrophobic, that we shouldn't have problems with cell permeability, that it might have some benefits over these very, very hydrophilic compounds that are involved in this interaction. But that remains to be seen. So I'm hoping, encouraging for the future. And I think that we were encouraged that we can decrease fibrosis in the asbestos model, and we just need to repeat it. We need to run it out a little longer. But the preliminary results were encouraging, and it does look like there might be some benefit as far as decreasing fibrosis in this asbestos treated. And we'd like to take this further and say, not just can we prevent the formation of damage, but like to be able to say, okay, now what happens if we delay treatment? So now we have damage. Okay, treat already damaged lungs. And can we get a reversion there? Can we actually get a benefit once the problem is already manifest, instead of just stopping the problem to begin with? So I'd like to acknowledge Dr. Catlin Toth. She's a postdoc researcher in the lab and conducted a large number of the studies in this experiment. Susan Kahn did a lot of the fibrosis models with Dr. Toth. Claudia Ruiz provided a lot of the biochemical and bioanalytical support. And Ted Kamenika and Diju are synthetic organic chemists and made the molecules for us. And even though they didn't end up working with the photoactivated ligands, they made several of them for us. It was an idea. It just didn't end up working out.